Development in humans and other mammals is more complex than frog development because the embryo doesn't develop as an independent animal. The egg of a mammal is much smaller than that of a frog or a bird, mainly because the mammal egg has very little yolk. Mammals provide their young with nutrients throughout embryonic development, so the egg doesn't need to contain a large supply of nutrients. As the cells of the human zygote divide, they develop into a stage called the blastocyst, a structure roughly equivalent to the frog or chick blastula. The cells are arranged in two layers. The inner layer will develop into the fetus. The outer layer will become the placenta, the organ of exchange between the mother and fetus. There is also a blastocele, an embryonic cavity we first saw in the frog embryo. The inner cells at this early stage can differentiate into any cell type. These cells are known as embryonic stem cells. Embryonic stem cells are important in medical and scientific research because they have the potential to grow into any type of human cell. In the future, stem cells might be differentiated to replace insulin-producing cells in the pancreas that are missing in a patient with diabetes, or other stem cells could differentiate into healthy heart muscle tissue that could be injected into a patient with heart disease. Some people believe that even an early-stage embryo has the capacity to grow into a human being and therefore feel it is unethical for cells to be used in this way. Others believe that the potential benefits for humankind are too great, and that it should be the parent's decision whether or not to allow embryonic stem cells to be used in medical research. In normal development, the stem cells and the encasing outer layer of cells which make up the blastocyst sink into the wall of the uterus. This process is called implantation. The outer layer of cells will eventually form the placenta. This layer thickens and extends further into the wall of the uterus. At the same time implantation is taking place, the same membranes we saw forming in the bird egg are developing in the human embryo. Here we see the amnion, chorion, allantois, and yolk sac. There are some modifications to the role of these membranes in the human embryo. For example, the human yolk sac doesn't contain any yolk. It eventually becomes the site of early blood cell production. The human allantois is incorporated into the umbilical cord, the tube containing the blood supply between the fetus and the placenta. The human embryo's waste products will be exported from the embryo to the mother, so there's no need for the allantois to function as a waste receptacle during the mammalian development. At the center of the developing embryonic tissues are the three germ layers that will develop into the embryo itself. These are the same layers that we saw in the frog embryo. As development proceeds during the first trimester of pregnancy, the placenta becomes visible as a disc-shaped structure. By the end of the 8th week, all the major organs are present, and the 5-centimeter-long embryo is usually referred to as a fetus. In a close-up view, you can see that the maternal and fetal blood vessels are not connected. However, they are very close to one another, so that oxygen and nutrients can diffuse across to the fetus from the mother's circulatory system, and waste from the fetus can pass back to the mother. In the second trimester, the fetus grows to 30 centimeters in length, and the fetus may show active movement. During the third trimester, the fetus grows to around 50 centimeters in length. Development of the human fetus concludes with birth. A series of strong rhythmic contractions of the uterus expel the baby through the vagina. The umbilical cord is cut, and the final stage of birth is delivery of the placenta.